Conventional science recognizes that all matter, each molecule of everything on Earth and beyond, oscillates at its own distinct frequency.
Can the amplified voice of Ella Fitzgerald shatter this glass?
The atoms that come together to form a molecule are held together with an energy bond that both emits and absorbs its own specific electromagnetic frequency. No two species of molecules have the same electromagnetic oscillations or energetic signature. By increasing the intensity of the harmonic frequency of the molecular structure of the wine glass, science can cause the glass to shatter.
Believe it.
This is a commonly accepted law of nature. So why can't other molecules be isolated and destroyed in the same way? Why can't the oscillation frequencies of killer viruses like cancer and AIDS be identified and destroyed? Why hasn't someone performed the research? Well, someone has. Royal Raymond Rife was a brilliant scientist who was born in 1888 and died in 1971. His studies at Johns Hopkins University led to his development of the technologies that are still commonly used today in the fields of optics, electronics, radiochemistry, biochemistry, ballistics, and aviation. He received 14 major awards and honors, including an honorary doctorate from the University of Heidelberg. During the 30 years that Rife spent designing and building medical instruments, he worked for Zeiss Optics, the U.S. government, and several private benefactors. Because Rife was self-educated in so many different fields, he intuitively looked for his answers in areas beyond the rigid scientific structure of his day. He had mastered so many different disciplines that he literally had at his intellectual disposal the skills and knowledge of an entire team of scientists and technicians from a number of different scientific fields. So whenever new technology was needed to perform a new task, Rife simply invented and then built it himself. In 1920, Rife began investigating the possibilities of treating disease with electricity. He discovered that each disease he studied had different electrical characteristics and started subjecting these organisms to different electrical frequencies. His first virus microscope complete, Rife performed tens of thousands of lab tests in an effort to isolate the microorganism that caused tuberculosis. Traditional scientific procedure requires the staining of samples to make them visible under the microscope. Unfortunately, the minuteness of the viruses made them impossible to stain with the existing acid-based stains. Rife conceived a method of staining the samples with light and began building a microscope that would enable the frequency of light to correspond with the electrical frequency of the microorganism under observation. His cancer research began in 1922, but it would take until 1932, after thousands of tests, that he was able to isolate the microorganism which he named the BX virus. By 1933, he had perfected that technology and had constructed the incredibly complex universal microscope, which is capable of magnifying objects 60,000 times their normal size. With this incredible microscope, Rife became the first human being to actually see a live virus. Until quite recently, the universal microscope was the only microscope able to view a live virus. Rife painstakingly identified the individual spectroscopic signature of each microbe. Colleagues of Rife reported his incredible patience, sitting at the microscope many times for over 24 hours straight without a break. Once he discovered the oscillation rate of a particular organism, Rife concentrated on refining his method of destroying it. He used the same principle to kill the virus as he had used to make the organism visible: light frequency resonance. By increasing the intensity of a frequency that resonated naturally within these microbes, Rife increased their natural oscillations until they distorted and disintegrated from structural stresses without harming the surrounding tissue. Rife called this frequency the mortal oscillatory rate, or MOR. Half a century later, doctors began using a like technology known as lithotripsy, where kidney stones are destroyed with high-energy shock waves. Identify the MOR of cancer cells, turn up the volume and destroy them without damaging the host organism.
As far back as 1920, I conceived the idea and the possibility of when The causative agent of malignancy, so-called cancer, would be discovered and found and proven that it would be caused by a microorganism. Of course, reception that I received that far back from the medical profession and scientists was nil. But I kept at the work, and I succeeded in eventually isolating a virus. First, I began sectioning tissues of every known type of malignancy. I sectioned over 20,000 of those, cut them down with a special microtome to very thin. Some of them were only a micron in thickness. I studied those under the microscope, and I eventually built my first high-powered microscope for the purpose of analyzing and checking those sections.
Rife found, through arduous experimentation, looking through the microscope as he applied various frequencies, that he was able to kill these microorganisms, proving his theory to be correct. In order to determine which frequencies would kill a particular organism, it was absolutely essential that he be able to see the organisms in a living state so that he could observe the effect of various frequencies in real time. Rife later called a frequency which killed an organism its MOR, or mortal oscillatory rate.
As an example, it's worked out, my theory was worked out on a basic principle of what we term coordinated resonance. Now we take and we put a slide of a certain chemical or material under the petrographical micropolariscope, which is that second microscope down there. I didn't build that, I designed it. Now, we have what we call a coordinated resonance, you see. We have a chemical here, we have a frequency. All right. We will take and change those to two tuning forks that's pitched to the C absolute. We put the other one down there, we strike this one, the other one resonates. It's coordinated resonance, owing to the chemical constituents. And we create that electronically, the same as we determine what material is under that microscope by the color and the index of light as we rotate it between the analyzer and polarizer. As the color index fades from one color to the other, the angle of refraction of that crystal and so on, we can tell. It's coordinated resonance. The same as I say, you take two tuning forks that's pitched to the C or an A absolute, you strike one, the other one resonates. So we're showing an electronic frequency through the tissues of the body that simply devitalizes the bacteria with no harm to normal tissue.
With that instrument, Arthur Kendall and I, working jointly in the Pasadena General Hospital, succeeded in isolating what we classify the first filterable form of bacteria ever seen. It was isolated from the Bacillus typhosus from cultures that Dr. Kendall brought from his laboratory in Northwestern University in Chicago. And we succeeded definitely in isolating a filterable form of that bacteria. Now these filterable forms are very minute in size. The smallest of all is the one from the BX, which we isolate from cancer. We have never made a positive claim that that is the causative agent of cancer. But that is the smallest, and it's less than a 1/20th of a micron in dimension and highly motile. We succeeded in isolating this organism, this BX, using a Kendall media. It's known as Kendall media. It's made of a desiccated pig intestine, dehydrated down where it requires